This video is Unit 3, Part 2, and Part 3. I'm going to kind of throw a curveball from my normal video routine. Part 2 and Part 3 are fairly short, and so I thought I'd combine them. Don't worry, I will still have a title screen to make sure that you're all taken care of there. Just wanted to show a quick picture of a receptor protein. Remember, proteins are made up of primary structure, secondary, tertiary, those sorts of things. Here's a tertiary structure, and this shows you that this shows the binding site and then how that binding site could affect the rest of this protein at the molecular level. And it just reminds us that proteins have shapes, or shapes determine their function. Oftentimes when I show you a protein, it looks something like this weird slipper here, but really what's going on is something much more chemical. And that brings us to the next thing. We're going to talk about a specific cellular pathway called the G protein pathway. This is the most common pathway used by cells. A couple of parts of this pathway that you need to be familiar with. First is the G protein linked receptor, fancy way for saying this is the place where the ligand is going to bind, and this is found on the plasma membrane of the cell. Again, not on the inside of the cell, but on the outside of the cell. All cells possess G protein receptors. This allows them to interact with, respond to their environment around them. The actual G protein itself is this blue blob here that has that GDP on it. We'll talk about the GDP in a second. This protein is essentially going to act like a relay protein between the G protein linked receptor, which interacts with the outside world, and this enzyme over here, which will remain unnamed because it could be any number of enzymes. And this bottom picture kind of shows you what's going on. Ligand, or the signal molecule, ligand comes in, causes this G protein to be activated, moves over here, activates this enzyme. There's a couple things to fill in the blanks here. Well, first of all, that GDP that is on the G protein is going to be exchanged by GTP. GTP is just like our friend ATP, except for it's guanosine triphosphate. And just like ATP, when guanosine triphosphate binds to something, it activates that thing. In this case, it activates the G protein. The G protein does one thing. It goes and talks to that other enzyme, whatever enzyme that is, and it's going to phosphorylate that enzyme, which we understand phosphorylation to be just the adding of that phosphate, almost like turning on an on switch, and that enzyme is then going to initiate some sort of cellular response or further the transduction pathway, whichever occurs. Important concept here, don't miss the forest for the trees. This is just a very simple relay system. Signal binds, G protein goes and says, okay, I got the signal, takes it to the enzyme finishes off the response. Very simple. Another type of simple, well, less simple than G protein, but still very simple, is called the tyrosine kinase receptor. It's called tyrosine kinase because of the tyrosines located in the activation regions here. Kinase should ring a bell. It's a signal that is being turned on, and so in this particular case, notice what's going on. You have two separate signal binding sites here. The ligands come in, they bind, causes this conformational shape change, which causes these two proteins to join together. Upon doing that, not just one activation site is activated, but all 6 of these activation sites are activated. And so in this way, 6 different cellular responses can be initiated. If you think of a process like repair or growth, you cut your finger, and what needs to happen? Well, several different proteins need to go into action. There needs to be a protein that stops the bleeding. There needs to be a protein that starts growing new cells. There needs to be a protein that gets rid of all the trash that's in the area. There's all these different things that are going to be made. And so a tyrosine kinase receptor is going to be the best and most efficient way to initiate several cellular responses with one signal molecule. Another type of signal or signal transduction pathway would be the ion channel receptors. Ion, because the ions are what is going to go through the channel. I showed you this one in a previous video. So you have the signal molecule here, The signal molecule acts just like a key to a gate. Upon being put in the gate opens, the ions diffuse through high to low, and when the signal molecule goes away, the gate closes. How does the signal molecule go away? There's usually an enzyme that breaks it down or whatnot. We'll talk about some specific examples of this, but this is particularly important in muscle cells and nervous cells. One more type of pathway is called an intracellular receptor. Intra means within, and so whereas we've been talking about signals being received on the outside of the membrane up to this point, an intracellular receptor is where the receptor is actually located inside the cell. There are certain kinds of molecules that can pass directly through the plasma membrane. Hormones that are steroids are a great example of this. Here's a picture of testosterone. Remember, steroids are lipids. The cell membrane is largely nonpolar because of the phospholipid bilayer, and so the steroid passes straight through the membrane into an intracellular receptor, which begins the transduction pathway and then initiates some sort of cellular response. So don't make this more complicated other than to say that pathway with steroids is on the inside of the cell rather than the outside. Now let's talk about a second category of messengers. These are called secondary messengers. They're relay proteins within the cytoplasm of the cell. The most common relay protein is called cyclic AMP, and they move about inside the cell. They're waiting to relay some sort of transduction pathway. Here's a picture of ATP, which you should be familiar with, adenosine triphosphate. Well, this particular enzyme, adenylyl cyclase, takes off those two phosphates. You have cyclic AMP, or adenosine monophosphate, and this cyclic AMP acts like a secondary messenger. What do we mean by that? Well, here's the simple G protein pathway that we talked about earlier with a secondary messenger thrown in. So G protein does its thing, and secondary messenger cyclic AMP is going to be a part of that relay process. That's one example. Another example of a secondary messenger is calmodulin. You see at the top of your screen, calmodulin. Calmodulin is important in the muscle contractions. There's a whole lot here, and I think it's important just to make sure and understand. We will talk about in another unit about this particular process, but the main thing that I want you to draw away from it is G protein. Notice there's the enzyme, some sort of cellular response, some sort of other secondary messenger, another secondary messenger, another secondary messenger, and you have this normal relay that's going to initiate some sort of cellular response. In this case, that cellular response is going to be muscle contraction. And so secondary messengers are a part of that relay system. Now for part 3. The first thing to talk about in part 3 is this idea of cascades. I drew this picture yesterday when we were lecturing, so I just decided to keep it. Here's a picture of some champagne glasses. Normally when people think of Cascade, they think of dish detergent. Well, used to. On the cover of Cascade dish detergent was a champagne glass because people think of things called champagne cascades or whatever they're called. The idea that you have a bottle of champagne and you are pouring it into, this is a really bad bottle of champagne, and you're pouring the liquid into this cup and it overflows into these cups and it overflows into these cups and so forth. And so as it does that, it's cascading down. Filling one cup is actually initiating the filling of many cups after that. Well, how do we translate this into cell communication? You have one signal molecule here, and through that one signal molecule, you have the initiation of many cellular processes. If it's a kinase cascade, then you're turning many cellular processes on. Think of this as amplifying the signal. There's one signal, but lots of responses are being amplified through all of these different relays that are going on. I think of this as like a call chain, which used to be a thing before group text, where if it was snowing outside and school needed to be canceled, the superintendent would call 2 people, probably the principals or something along those lines. Those two people would call two people each. Those four people would call two people each. 8, 16. It gets real big real quick. And so in just a few minutes, the entire school district, particularly the teachers, know, hey, you don't have to come to school today. And so that was a way that it was done in the past. Well, in this particular case, you have one signal molecule, the superintendent, who calls off school. And then from that one call, many different relay protein or many different relay processes are being made until so many different people know. The opposite can be said with phosphatases, which just turn off a bunch of processes. The same as the kinase, where you have this amplification of a signal, but it's just turning it off. This picture shows that amplification, where you have a single molecule with reception can initiate a response that could cause 10 to the 8th molecules being activated because of those responses. So this is a picture of that. You have some sort of response, this phosphorylation cascade turns on many different things. The response is happening. Again, don't lose sight of the forest for the trees. One more thing is this idea of going from simple to complex. So a simple example of a cellular pathway is this idea of a scaffolding protein. Why am I calling it a scaffolding protein? You have this direct stimulation from the signal molecule. Multiple relay proteins are being turned on at the same time because of this one scaffolding protein that has 3 different binding sites. Well, we looked at something just a few minutes ago on something that is a more complicated version of this. This tyrosine kinase receptor, which can turn on 6 different processes at once. And so imagine this being a simpler, less complex version of this, or this, the tyrosine kinase receptor, being a more advantageous than this. And so this concept of evolution changing over time, we understand evolution to weed out the less efficient systems. These systems still exist, and so we still see these very simple scaffolding proteins and other organisms, but the organisms that are most efficient, most evolved, if you will, are the ones that have these more complicated systems like the tyrosine kinase receptor. Use this as an example of a larger concept rather than a specific. It is a specific example, but understand as organisms evolve, these processes are going to get more and more complex.